Hi, welcome to Mr. Leek's free science tutorials. This is a very, very common question that comes up right the way through GCSE biology. And basically what it is, it's describe how I would test a cheese sandwich for sugar, starch and proteins. So it's really what it's doing is it's asking for the food tests for sugar, starch and proteins. In your exam, make sure you don't just answer one of them, give one test, make sure that you answer all three of them. Now, these are then the three tests. Now, there are others, for example, a fats test, but these are then the three most common ones that are ever asked about. Sugars, particularly glucose, is the Benedict's test. And what it does is it has a variety of colours depending on how much sugar there is actually present. It starts off at blue. That is the starting colour of the Benedict's reagent, right, for the Benedict's test. It then goes green, yellow, orange, brick red. And however, or whatever the colour is, depends on how much sugar there actually is. So it says here, green is traceable. High is brick red. Right, so if the actual question says, what is a very positive test for the Benedict's test, you would say brick red. The second one is test for starch. So typical thing for starch is bread, potato. If you put iodine onto bread or potato, what it does, it turns black immediately. So starch test is iodine and it turns black. For proteins, it's the Biuret reagent. Sometimes it can come as Biuret 1, Biuret 2, or a combination one. And if there is protein present, it goes mauve or purple. So basically mauve, it's kind of like a really nice, palish kind of purple colour. So, back to the question. Describe how I would test a cheese sandwich for sugars, starch and proteins. I would test a cheese sandwich for sugars using the Benedict's test, and it would go from blue to brick red depending on how much sugar is present. I would test for starch using iodine. If there is starch present, it would turn black. I would test for protein using the Biuret reagent. If there is protein present, it would turn mauve or purple. They are then the three main tests on how would I test a cheese sandwich for sugar, starch and proteins. Welcome to Mr. Leek's free science tutorials. The question in this video is, how are carbohydrates, proteins and lipids, fats, broken down in the digestive system? Where are the enzymes made and what are the food groups broken down into? So firstly, right, let's think about it. You eat your burger, your McDonald's, whatever it may well be, it's going to contain carbohydrates, proteins, lipids. The question is asking, right, where are the enzymes made and what are the food groups broken down into? So here's your digestive system. The mouth and the teeth, whole job of that is to maybe add lubrication and break it up into smaller pieces. The esophagus is a muscular tube that takes the food from your mouth all the way down to your stomach. The stomach then contains hydrochloric acid, which main role is to sterilize the food to make sure that there is no bacteria or infections or other microorganisms in there, and it has a pH of approximately 1, so that is a very, very strong acid. When the food's been in the stomach and it's churned around and it's broken up, it then comes out of the stomach into the small intestine, which starts round about here, straight after the stomach. At that point, other things are then put into the digestive system. The pancreas puts enzymes in, and what the pancreas looks a bit like a leaf. You can't really see it particularly very well on there, but it's a bit like a leaf shape. You've then got the liver and you've got the gallbladder. Both of those are then introducing different things into the digestive system. The stomach pH is about 1. If that came out of you, right, then I assume or I guess it'd be quite uncomfortable at pH 1 coming out of your body. So the gallbladder, right, and other areas around here, right, produces the bile, right, and that bile then goes in and neutralizes the stuff that's coming out of the stomach. It then goes into the small intestine. The small intestine, huge, looks a bit like the brain, right? All the different bends and movements and stuff like that. It then comes out here into the large intestine. This little fingery little bit here is the appendix. It's not labelled on here, right? But the appendix is then a vestigial organ that we then don't use anymore. It used to be used for digesting grass and other hard to digest substances. The large intestine, right, then goes all the way round, and the main role of the large intestine is to absorb water. So basically the stuff that's in your intestine gets thicker and thicker and thicker and thicker until it comes out of the anus. Now, the actual question itself isn't asking about the organ systems. It's asking about how are carbohydrates, proteins and lipids broken down in the digestive system? Where are the enzymes made and what are the food groups broken down into? Now, food is basically broken down by enzymes. You've got carbohydrases, break down carbohydrates. Proteases, break down proteins. Lipases, break down lipids or fats. And I always remember that where people on the news have liposuction. Liposuction is where a tube goes into the stomach with a few other processes and the tube then sucks out, right, lipids, lipo, and that is then fat. The last one on here, even though it's not an enzyme, bile is incredibly important in the whole process. So, carbohydrates, like your sugars, made in the salivary glands, the pancreas and the small intestine. Most common one that people hear about, salivary glands is salivary amylase. So when you eat your piece of bread, a good little experiment is you get a piece of bread in your mouth, chew on it, chew on it, chew on it, chew on it. The salivary glands or the salivary amylase break down the carbohydrates into glucose and the bread eventually gets quite sweet. What the carbohydrates do, they break down the carbohydrates to simple sugars. E.g., amylase breaks down starch to glucose. But the key to it is they are made in the mouth, in the salivary glands, the pancreas and the small intestine. Proteases. Proteases break down proteins, obviously. Main place in which proteins are digested are in the stomach at a pH of 1, and in the pancreas, or not in the pancreas, but enzymes, proteases are then produced from the pancreas. They break down proteins to amino acids. So a protein is basically a massive, great big long chain of amino acids. Lipases, your fats, right? The enzymes lipases are made in the pancreas, right? And what they do is they work in the small intestine. Lipids and fats are actually quite difficult to digest. Lipases break down lipids, fats to glycerol and fatty acids. So you've got a lipid, it gets broken down into two things, glycerol and fatty acids. Now bile, as part of the digestive system, is a bit like a liquid, right? It's like a yellowy sort of straw-coloured liquid, and it is very, very alkaline. It's made in the liver and stored in the gallbladder. And what it actually does, and this is then key terminology, it emulsifies the lipids to increase the surface area. If you think about if you put a greasy frying pan, right, into your washing up bowl with just water in it, the grease just sits on the surface. As soon as you put in fairy liquid or any other detergent, what happens is the little globules of fat break up. That is emulsify. And that does is increases the rate of lipid breakdown by lipase because the surface area is increased. It also changes the pH to neutral for lipase to work. Lipase and lipids, lipase doesn't work in acidic conditions. So if we then look at both of these diagrams together. Okay, so carbohydrates. Carbohydrates are made in the salivary glands, the pancreas, and then the small intestine. Their role is to break down the carbohydrates to simple sugars, for example, starch to glucose. Proteases are made in the stomach and in the pancreas. They break down proteins to amino acids. Lipase is made in the pancreas and it works in the small intestine. So basically the pancreas produces the enzymes and then they're gradually broken down as they go through the small intestine. Lipases break down lipids to glycerol and fatty acids. The last part is bile. It's made in the liver. It's stored in the gallbladder, which is that small green bit. And its role is it emulsifies lipids to increase the surface area, to increase the rate of lipid breakdown by lipase. And it also changes the pH. So, how are carbohydrates, proteins and lipids broken down in the digestive system? Where are the enzymes made and what are the food groups broken down to? So, carbohydrates are broken down by carbohydrates, made in the salivary glands, the pancreas and the small intestine, and they break down carbohydrates into simple sugars. Proteases are made in the stomach and the pancreas. They break down proteins to amino acids. Lipases are made in the pancreas, work in the small intestine, and they break down lipids and fats to glycerol and fatty acids. Bile is made in the liver, stored in the gallbladder, and it emulsifies lipids to increase the surface area, and it changes the pH to neutral for lipase to work. Welcome back to Mr. Leek's free science tutorials. The question now is, explain how a vaccination works. Now this is obviously a very topical subject at the moment with coronavirus and COVID infections being all over the place, and a lot of big companies are trying to get a vaccine to prevent people catching it. Now firstly, what is a vaccine? A vaccine is used to immunise a large proportion of the population to prevent the spread of a pathogen. So a pathogen is a microorganism that causes a disease. Large proportion of the population, if the large proportion of the population is immune to a disease, then it reduces the possibility of it being transferred. And that was then what was talked about right at the very start of this pandemic, which is herd immunity. If there are a lot of people who are immune to something, then very few people will then actually catch it or catch the actual disease itself. Now, a vaccination. Explain how a vaccination works. Now, what a vaccination is, it's usually a small amount of dead or inactive form of the pathogen. Now, how these scientists do it, right, is absolutely incredible. But what they'll do is they'll get the pathogen, and what they'll do is they'll kill it, right, or they will somehow get an inactive form of it. And what they'll do then is they will inject it into the person, which is called this, the first injection by the pathogen. And what your body actually does is the white blood cells detect pathogens in the vaccine. So you have that vaccine injected into you. The white blood cells in your body travel around the body, and eventually what they do is they find the pathogens, which are usually dead or inactive. The white blood cells release antibodies, and those antibodies then bind to the pathogen, right? What they do is they bind to the outside of it, because all pathogens have a certain shape around the outside. And what they do is they then make the actual pathogen itself inactive, and then they can get rid of it. The good thing is, and then what that is, that's then the vaccination itself. If that person then gets reinfected by the same pathogen, the white blood cells find the pathogen that's in the blood, and the antibodies then are made much faster in larger amounts, and they get rid of the disease, the pathogen, much, much quicker. So, explain how a vaccination works. Firstly, I would do in this question is I would say what a vaccine is. A vaccine is used to immunise a large proportion of the population to prevent the spread of a pathogen. A vaccination works by injecting a small amount of dead or inactive form of the pathogen into the human. The first infection by the pathogen or the vaccine, what happens is the white blood cells detect the pathogens in the vaccine. So it goes into your arm, the white blood cells detect it, antibodies are released into the blood, and those antibodies then make the pathogen inactive. But because it's inactive anyway, you weren't actually going to get the disease. It's kind of a bit like the shape that they're detecting. If you're unlucky enough to then get the disease, you're reinfected, what would happen is the white blood cells detect the pathogen, and the antibodies by those white blood cells are made much faster and in larger amounts. And so basically what you do is you get that new infection and it doesn't actually become the disease because you're then immune to it.